The Epigenase Universal SIRT Activity Inhibition Assay Kit provides a fast and convenient way to measure the activity or inhibition of total sirtuins in your samples. Unlike traditional methods, the Epigenase kit directly detects enzyme-converted deacetylated products, giving you accurate sirtuin activity results. The kit is compatible with cell and tissue extracts as well as purified SIRT enzymes, allowing you to assess both in vitro and in vivo inhibitor effects. In this assay, an acetylated histone SIRT substrate is stably coated onto microplate wells. Active SIRTs bind to the substrate and remove acetyl groups. The deacetylated products are then specifically detected with a high-affinity antibody and quantified colorimetrically using a standard plate reader. The kit includes all essential components needed to perform the assay. To begin, prepare the standard curve by diluting the SIRT assay standard with assay buffer to create a range of standard concentration points. Arrange the required number of well strips in the plate frame. Next, add assay buffer to each well. Load your prepared standards in duplicate to establish the standard curve. To the blank and sample wells, add the specified amounts of SIRT substrate and cofactor. Also add the specified amount of HDAC inhibitor to the sample wells. Then, load your samples into duplicate wells in the same manner as the standards. Cover the plate with the provided adhesive film. Incubate at 37 degrees Celsius to allow the enzymatic reaction to occur. When incubation is complete, remove the solution and wash the wells with wash buffer. Add capture antibody to all wells. Cover and incubate at room temperature. Following incubation, wash away unbound capture antibody, then add detection antibody. Cover and incubate again at room temperature. After a final wash to clear out residual detection antibody, add developer solution and monitor as a blue color develops. When the desired intensity is reached, add stop solution. The color will change from blue to yellow, and the plate is ready for measurement. Read the absorbance at the recommended wavelength. Plot the standard curve using the OD values of the standard concentration points and calculate the enzyme activity in your samples. With the Epigenase kit, you'll achieve fast, sensitive, and dependable quantification of sirtuin activity or inhibition. For more information, visit epigentech.com or contact your local distributor.